Okay friends, now in this video tutorial we will be talking about the renal anatomy. In this series of videos we are going to talk about the different part of the urinary physiology or urinary system. Okay, and this is the first set of videos where we talk about the anatomical structure of kidney and the unit of kidney, that is nephron. And we know that nephron plays a vital role for making the urine concentrated as well as absorbing vital elements from the excretory juice, which we call ultimately released as urine. So let's talk about the urinary system overview, that is the anatomy. The goals for our learning is to review the anatomy of the urinary system, particularly the kidney, to examine the vascular and tubular portions of the nephron, to compare and contrast the specialized cells of the tubular epithelium, and to review the unique structure of the juxtaglomerular apparatus. We'll see all of them, and we'll also see the structures associated with the function. So we'll try to figure out what are the roles played by these structures. Let's move. If you look at the urinary system, it is composed of paired kidneys and ureter. Let me take a color so that I can illustrate all this drawing. Okay. The major component of our primary urinary system here, it is with this a pair of kidneys. Okay, and we have a ureter. This is connecting the kidneys ultimately to the urethra. Urine is produced in the kidney and then drains through the ureters ultimately to the urinary bladder. It stores there for a while, then due to the contraction of the sphincters there, it's released out. Okay, now if you look at this picture, it will give you an idea about the position of the kidney. Kidney is like bean shaped organ and they are placed slightly above your waist. This is the range where the kidney is placed, and it's mostly placed to the back side of our body anatomy. Okay, it's not in the front, in the back. Now if you go to the next page, it will tell you the idea about the exact position. Here you can see the exact position of kidney in a human body. It's just above the waist, and two kidneys are not at the same horizontal level. Okay, they have a slight change in the kidney. The right one is slightly lowered down compared to the left one. If you look at the external structure of the kidney, this bean shaped structure is embedded in the fatty adipose capsule. Okay, and these kidneys are retroperitoneal, that means lying against the dorsal body wall of the upper abdomen. Okay, and they also have another special gland structure present on top of both the kidneys, which is not drawn here but I am drawing now. This is known as adrenal gland. Okay, which is a part of the endocrine system. It is it normally present in the top of the kidney. It remain attached there, and this adrenal gland used to secrete different hormones. Okay, now if you look at kidney, if I zoom in other structure, if you look at here, you'll see that several structures enter or exit this concave region of the kidney. Okay, and these structures are mostly vein and other nerves. They are known as renal vein or renal nerve and renal artery, because you know kidney's job is to make the urine. So while the urine is being produced, what is urine in a sense? You know, our body produces all those excess materials, toxic materials, which are converted to urea. Normally it's made as ammonia, but ammonia is much toxic and more dangerous form, so we convert ammonia into urea, and once urea is produced, that urea gets transported through this region of nephron. And what blood is doing here, blood circulation is required, because even though we produce the urea and maximum part of it is water, but still that sometimes contains sodium or glucose or any other important mineral or nutrient substances. It may present in that excretory juice, you can say. Then in that case, blood needs to reabsorb those things back so that body can utilize them for the other work, useful work. That is the reason it should be like it make a network of vein and arteries together with the help of this renal vein and renal artery. On the other hand, there is this renal nerve which is also engaged in interaction with the same region. Now, as we are talking about the blood supply of the kidney, you will understand when the renal vein is removed and the kidney is shown in frontal section, you can see the deeper branches of the kidney. The branching from the renal artery are segmented to the lobular arteries. Okay, so this is the renal artery. It start making a branch called interlobular blood vessels, and then finally those interlobular blood vessels are getting further divided and further segmented, known as cortical blood vessels. Okay, because if you look at the structure of the kidney from outside, we can say it's made up with two sections normally, renal cortex and renal medulla. The cortex part is this surrounding layer. This is the cortex part, while the inside this region is known as the medulla. Okay, so whatever structures we see in this cortex region, we call them cortical structures. Okay, so it's if it's a part of nephron, we call it a cortical part of nephron, or in this case the vessels, cortical blood vessels. Now if you look at the internal structure of kidney, look here at renal cortex, the one that I just talked to you about. The outermost layer is known as the renal cortex. It contains about 1 million nephrons, which are simply the filtering unit of the kidney. So if you look at all this cortex and you see the structure of nephron or how nephron is arranged and organized through this cortex. Okay, and nephron have several different structural elements and units, but if you look at here, you will find out that these nephrons, especially that the primary unit or maximum part of the nephron is present in this cortex region, and the nephron region that is present in the cortex is known as cortical nephrons. Okay, which is completely within cortex, but there are some other nephrons, some part of which is in the cortex, some part is inside the medulla. Okay, so that is juxtamedullary nephron. So there are the two types of nephrons available. If you look at this picture, you will see that one nephron can be straight present in the cortex, that is cortical nephron, but this one is some part is in cortex, some part is in medulla, known as juxtamedullary nephron. The other part of this nephron, other part of the kidney is renal medulla, that is the middle layer. This is the layer known as medulla. And in this case, if you look at here, this medulla is mostly made up with this triangular structures. Okay, it's something look like the pyramid, we call them. This pyramid look striated because the parallel bundles of the ducts carrying urine from the nephrons and finally dragging to the ureter. That's why you see kind of striped structures in this medulla triangles. And the third part of this kidney is renal pelvis, which is this one. This is the renal pelvis, just like a funnel. In this picture it will be much clearer. This is the renal pelvis, structures like a funnel, which funnel the urine and start to enter the urine through this renal pelvis into the ureter. Okay, and then finally it will reach the urinary bladder. So that is the structure of the kidney. Now if you go and look at the structure of nephron, because that is very, very important, because the nephrons are the actual unit structures that helps in filtering and production of the urine properly. Okay, so let's look at that. Okay, the structural point of view of nephron can be from two different sides. We'll talk it from two different angles. First one, we will talk from the angle of the blood vessels associated with the nephron. Then we also talk about without the blood vessels or the tubular form of the nephron. So if you look at the associated blood vessels, blood entry is very important as well as exit. So here we see the entry point of the blood, here we see the exit point of the blood. Because you see here, this is a head-like structure of a nephron that is known as Bowman's capsule, where there is the transfer of all this high pressure blood is initiated, and due to that process water and other things are transferred inside the nephron. So blood entering the kidney through the renal artery flows first into the segmental arteries and then into the lobular arteries. You know that? First it will entry into the interlobular segmented artery, interlobular artery. Okay, that's how they are transferred. And then finally it enters the arcuate arteries. The small interlobular arteries and the smaller arterioles are also present surrounding the cortex region. We saw that earlier. And this is the region when we find this Bowman's capsule. This Bowman's capsule have inside, there is a bunch of arterioles tagged inside, known as the glomerulus. Okay, so that is how ultimately blood reaches into a capillary bed, into the glomerulus. This glomerulus is a very, very important structure, important arterial structure through which the blood is transferred. Okay. Now here this glomerulus is efferent arteriole, and if you look at here, the afferent arteriole is larger in diameter than the efferent arteriole. If you look at the structure, I don't think this picture is clearly representing that fact, but actually if I draw the structure, let's say this is the Bowman's capsule and this is the glomerulus, and what you will find here is that the afferent arteriole, the diameter for the afferent arteriole is much bigger compared to the diameter of efferent arteriole. So what happens? The blood pressure increases in the efferent arteriole, and that is important for the separation of the components from the blood, for the purification of the blood. So blood passes from the efferent arteriole into the peritubular capillary and and finally to the vasa recta. We will talk about that. From there the blood drains into the interlobular vein, flows into the acute vein and enters to the interlobular vein. Okay, and finally to the renal vein. Okay, if you look at here, this is the structure representing, this is the afferent arteriole, and you know the blood is flowing through this afferent arteriole and then finally going through the glomerular region, and then finally it goes back to the efferent arteriole, and you see the diameter is already varied, it's changed. Now this cup shaped structure we talked about as Bowman's capsule, water and solute pass from the blood into the glomerular capsule or Bowman's capsule and then flow into the proximal convoluted tubule or PCT, which is this one. And now focus on this picture. Blood start flowing through this glomerulus and then finally taking entry to the and all those solutes and water are taking entry from this glomerular into this first part of the nephron known as proximal convoluted tubule, and then it's transferred from there to another very thin diameter structure known as loop of Henle. Then finally it will come back to the distal convoluted tubule or DCT. So nephron, if you look in a whole, have several fragments. Starts with Bowman's capsule where they have glomerulus, then it has the proximal convoluted tubule, Henle's loop, distal convoluted tubule, and finally the collecting duct, which is collecting all these urine materials, and finally they are filtering it through the renal pelvis and enter the urine into the ureter. Okay. Now if you look at here, after the water and all those solutes which are taking engulfed like in this Bowman's capsule, they take entry to this proximal convoluted tubule. After many loops and convolutions, the tubule straightens out and fluid flows down descending structure. A thin descending segment known as Henle's loop or loop of Henle. Okay, and normally in many cases this Henle's loop is present in the medulla region of the kidney. Then again it will start going up, known as ascending limb, because we are going up afterwards. And this ascending limb is little thicker compared to the descending limb. Okay. Then from this loop of Henle, the fluid enters into the twist and turns, known as distal convoluted tubule. Then finally emptying all this material through the collecting duct. Now this is the overview of a nephron structure. Now if you zoom into one of this Bowman's capsule, this is the region, we call it as a renal corpuscle. Okay, so let's look at renal corpuscle and the structure of renal corpuscle. It contains glomerulus with its larger incoming afferent arteriole and smaller outgoing efferent arteriole. It is nested within a glomerular capsule, something like the first thrust into the balloon. Okay, this normal structure, the visceral layers of the glomerular capsule is made up of specialized cells known as podocyte. If you look at here, this is the structure. If you look at carefully, you will see there are some cell drawn here, elongated cell with nucleus, which is these dots surrounding this arteriole over there. These cells are known as podocytes, specialized cells. Okay. These podocytes surround the permeable capillaries. Between the visceral and parietal layer of the capsule, there lies a capsular space. This is the capsular space present between the capillary and the wall of the parietal layer of the glomerular capsule. So there is a space, and through that space there is also proximal convoluted tubule starts. Okay. Now if we zoom in this structure of podocytes and how they are surrounding it, you will see an important feature. These podocyte cells also have cytoplasmic extensions. This cytoplasmic extension helps them to grab every single inch of or every single centimeter, square centimeter region of this capillary, right? If you look at here, you can easily see. So that is very important because these podocyte cells also have this slits, filtration slits. With the help of these comb-like structures, or it's also known as the foot processes of the podocytes, they can help to transfer the components, which is water and solutes, from this Bowman's capsule region to the proximal convoluted tubule. Okay. This porous, another thing you can see, there are penetrations between these endothelial cells in the capillary, and these penetrations are important. Why? This porous basement membrane encloses capillary endothelium. Surrounding that basement membrane is this layer of podocytes, right? So they have this leg process or foot process like structures, right? So what happens, whenever substance being filtered must pass first through this all these fenestrations and through the basement membrane, and finally going through the filtration slits that are produced here by the podocytes. So that will properly filter out all the solutes that we need to filter them out. So you know, this is normally have these pores, fenestrations normally means pores. So through the pores all those water and contents will come out, but as the podocytes are covering the complete capillary structure and form these filtration slits, not all the molecules are able to pass this region. Only a few molecules will be able to pass, and the other one are retained in the blood. So together the capillary endothelium, the basement membrane and the podocytes make up the filtration membrane. It's known as a filtration membrane. Three of them things work together. So if I make a cross section of that, how it will look like? It will look something like this. This is the first section, that is the capillary endothelium. Let's say it contains all those pores. Okay, and then we have the basement layer. This is the basement layer. And then finally on the top of this we have the podocyte. So let me change a color, let me take another color. And finally we have the podocytes. So this is how it's arranged. So whenever a solute or any other molecules coming out from the pore, they need to again go through the basement membrane, then finally through the slits mediated by the podocytes to finally enter into the proximal convoluted tubule. So it's a several round of constraints and filtration units that are produced there. Now let's talk about the cells of the proximal convoluted tubule. Remember we talked about the different segments of nephrons earlier, and I told you like nephrons are containing three different types of tubules: proximal convoluted tubule, loop of Henle, and distal convoluted tubule. So if you look at the proximal convoluted tubule and the structures, you will see that this is simple cuboidal cells that actually make the structure of proximal convoluted tubule. If I draw it, let me draw this structure. We take a color here, it's not red, definitely we take a black color. So it's mostly all those tubular cells, but all these cells, they contains microvilli, okay? And if I draw something like this, it will look like this. This is how the cells are arranged and organized one after another, and they're linked with each other, okay? So this is how the cells are linked. This microvilli, which has a simply extension of the cytosolic domain or content. This microvilli greatly expand in the surface area of the luminal membrane, adapting it well for the process of reabsorption, because the job of this three tubules, that is the filtration job, is only done by the glomerulus and the glomerular apparatus in the Bowman capsule region. But the second part of the urine production is the reabsorption of the required minerals and nutrients. Because at the very beginning of the urine production that we saw, that is a filtration step that we saw, in the filtration step, all the materials that can take entry through that slits made by podocytes can pass. So we have wanted materials in our solutes and water, both of them, and we also have unwanted materials. Unwanted materials can be some say sodium, potassium, those are the minerals, those can also slip through that slits, okay? So the second part of the urine production job of nephrons, it's simply to take all the unwanted materials out from that urine. Unwanted means normally the products that we don't want in the urine, which are important products for our body. So we take up those products inside, and that is the job of all these three convoluted tubules and two convoluted tubules and the Henle's loop. Now if you look at very clearly in the structures between them, there are tight junctions between the adjacent cells. This is where the cells are kind of stitched and no room between the cells present at that stitching line. These are the tight junctions. These tight junctions between the cells permit passage of water but limit the escape of large molecules from the tubular lumen, which is this side we can draw, from the tubular lumen into the interstitial space, okay? It will prevent that. And the highly folded basolateral membrane of the cells contain numerous integral proteins involved in the passive or active transport of these substances between the intercellular and interstitial spaces, which is present here. This is also highly folded, as you can see. These are not microvilli, by the way, but this is also a folded cell membrane. It's not a simple structure of the cell membrane, and this kind of folding is also increasing the surface area. But the job is not reabsorption here. In this case especially, the job is the presence of different transmembrane proteins that will help them to transport different necessary solutes and molecules across the interstitial, like intracellular and the interstitial space between them. And they also have, all these cells also have numerous number of mitochondria. If you look at the structure here, it will be much clear. You see highly folded plasma membrane in one side, microvilli in other side, and all these cells carry a lot of mitochondria, right? And this huge number of mitochondria provide the chance of producing many ATP, which is necessary for these active transport processes that will go on in this highly folded region of the plasma membrane. And the key feature of this cell is that they are highly permeable to water and many solutes. So they are highly permeable to water and many other solutes. That is the specialty of these cells. Now if you zoom in and look at the structure of the cells that are present in loop of Henle, you know, there are two parts, two sections of the loop of Henle. There is a descending limb, another one is the ascending limb. The descending limb is thin, though in this picture I haven't drawn too much thin, but still compared to both of them, this is the descending limb, the thin loop of the limb, thin loop of the Henle, and this is the ascending limb of Henle. This is the thicker version of the Henle's loop structure. If we look at here, this structural difference is also playing a vital role in the separation of important nutrients and solutes from this excretory juice. The cells in the thin segment of the descending loop of Henle are simply squamous epithelial cells, as you see. You know, squamous epithelial cells that are present in our many part of our body. It's simply the epithelial, the simplest form of the epithelial cells, just like a scale of fish. This is the structure. And these cells lack brush borders, which reduce their surface area for reabsorption. So these cells continue to be permeable to water. They possess relatively few integral proteins and function as active transport molecule for the reabsorption of the, mainly reabsorption of the water, because solutes are generally unable to pass through these regions because there are very few transmembrane ion transporters or other proteins. Okay, so it's responsible for the water intake. You see here the structure of this thin section of the Henle's loop, and if you look at here, these are all those basolateral membranes and the interstitial fluid that is drawn here, but these are the squamous cells that are linked with each other, and they have a gap in between them through which also water can easily pass. Okay, now in this case, the key feature of the cells is that they are highly permeable to water, but not at all permeable to solute. Okay, remember the proximal convoluted tubule we saw, permeability for water is very high, permeability to solutes are also very high. Here permeability towards water is high, very high, but permeability for the solute is very low. That gives us an idea that this region of the Henle's loop is responsible for the uptake of water or reabsorption of water. So that is the reason, if you see the desert animals, this Henle's loop is quite longer. They have a large long Henle's loop because Henle's loop is designed in a way to retain maximum amount of water in those individuals, because those are desert animals, they need to retain water. Now if I look at the cells of the thick ascending loop of the Henle, and we'll also see the structure in the distal convoluted tubule. Okay, if I look at here, this type of cell, distal convoluted tubule structure, and it's mostly the thick part of the Henle's loop and the distal convoluted tubule, they kind of have very similar structural features together. The epithelia of the thick ascending loop of Henle and the early distal convoluted tubule are very similar. They are composed of this cuboidal cell. If we look at here, what are cuboidal cells? But These are simple square kind of structure of the cells, but they also have this folding of the cell membrane, and they have several structural differences compared to the cells of the proximal convoluted tubule. For example, these cells have fewer and very smaller microvilli. That's what we talked about. Very small. If you look at here, these are microvilli that we are seeing. But they are very small, and the number is also very few. Right? In addition, the luminal membrane is covered by a glycoprotein layer. So if you look at here, this is the luminal membrane, and that is covered by this glycoprotein layer outside. So very small and few microvilli, luminal membrane covered by glycoprotein layer. The basolateral membrane is very much similar to the proximal convoluted tubule, because here you can see the same structure while the cell membrane is folded, and we also see the cells are linked with each other, and there is a tight junction present towards the lumen, but it's open towards the interstitial fluid. Those are the regions it's open. Okay. Now the key feature of this cell is that they are highly permeable to solutes, and particularly sodium and chloride, but they are not well permeable for water uptake. So for water, very less permeable, but for solutes, very well permeable or highly permeable. That is the feature of this distal convoluted tubule, which is early distal convoluted tubule, because you know the Henle's loop is bending, and once it's bending, it finally will make the distal convoluted tubule. So the structure of this start point of the distal convoluted tubule and the ascending limb of the Henle's loop are mostly similar structures. Okay, just the diameter varies a little bit. Now finally we'll talk about the cells of the collecting duct. Okay, because you know these are the constituents of a nephron, starting from the glomerular apparatus, then we go to the proximal convoluted tubule, loop of Henle, then distal convoluted tubule, and then finally the collecting duct. So if you look at here, collecting duct, the cells in collecting duct and the late distal convoluted tubule kind of have similar structure. And here you see there are two types of cells present in this kind of collecting duct region, you can say, and these are also cuboidal cell. So similar structure based on the layer. Okay, but the first type of cell that we see here known as principal cell. These principal cells have more numerous, the number of these principal cells are huge, and they have a few microvilli. If you look at here, the microvilli is very few, and they also have a very few basolateral fold. Though they have this basolateral fold, this is the basolateral fold, these are the microvilli, but both the microvilli as well as basolateral folds are very low, very less frequent. Now these specialized cells respond to certain hormones that regulate the cell's permeability to water and solutes. So whenever there is a hormone release, they are designed in a way that they should respond in release of the hormone. Okay, and the key feature of this principal cell is that their permeability to water and solute is completely physiologically regulated by hormones. So earlier, whatever structures we saw, like distal convoluted tubule, loop of Henle, proximal convoluted tubule, all these structures, they have a fixed array of whether they will take water or solute. But in case of collecting duct, it's not mentioned from before. They are under maintenance by the hormone regulation. So that is a regulation our body controls based on the hydration state of our body. If our body is dehydrated, then obviously body will try to retain water, so they will release specific hormone to give the signaling of retaining water and not uptaking water. So our urine will be much concentrated and very less water will be there. While if our body is well hydrated, then the proper amount of water should be released. Now, if we move to the second type of cells known as intercalated cells. Intercalated cell. These cells actually, when the body acidity increases, these intercalated cells secrete hydrogen ions into the urine to restore the acid-base balance of the body. That is the actual function of this intercalated cells. The number of intercalated cells are comparatively less with the principal cells. Okay. The key feature of this intercalated cell is that their secretion of these hydrogen ions and balancing the acid base condition of our body. Okay. And these are the structures that we saw mostly. These all collecting duct structure that we talked till now are the part of mostly cortex, but some part of this collecting duct is also present in the medulla, and it should present in the medulla, because finally when all those solutes and water are reabsorbed and stuff is done, then the urine, we call that material urine, and that can take entry into the ureter using this collecting duct, right, through the renal pelvis. So what we see here, the structure that we find out, the structure of collecting duct that is present inside, mostly inside the medulla region, they have slight variations. Now in this medulla regions, the principal cells are mostly cuboidal in shape, just like this one. The luminal and basolateral membrane are also relatively smooth compared to this collecting duct that is present in the cortex regions. And the key feature of the cell is that they are also hormonally regulated and permeability to water and urea. Okay, because this is the region through which the urea will pass. So let's look at the summary of the anatomy that we've learned now. The urinary system is composed of the kidneys, ureters, bladders, and urethra. The kidneys is composed of three regions: the renal cortex, medulla, and pelvis. The function of the urinary unit, that is kidney, is a nephron. It's composed of a tubular portion and associated blood vessels. Some part of this nephron is present in the cortex, some part can be present in medulla. Now each region of the tubular portion of the nephron depends on the unique feature of its epithelial cells to carry out its function. Now all the segment of this nephron is mediated or structured by these epithelial cells. Some part of the epithelial cells help to filter the blood for the necessary solutes and water molecules that will make the urine, and some other parts of the epithelial cells that are present in proximal convoluted tubule, loop of Henle, and distal convoluted tubule will ultimately guide to transfer that urine into the ureter. So that in a sense is the anatomy review for the urinary system. We'll see an animation for that, and we'll also see the functionality of the nephron and how exactly nephron mediates the function of urine release. So stay tuned and watch this video. Thank you.